Hey everyone, my name's Jackie. I'm a second year student in GPN. Never done one of these before, so bear with me. So I'm in the Economo Lab, and we study how the brain controls movement broadly. And so something you probably heard about GPN is that it's kind of interdepartmental. So the Economo Lab is in the biomedical engineering department. So there's students from GPN, like myself, and there's also students from the BME grad program. So it's kind of like an interdisciplinary lab, which is really cool. So you have people from different, different backgrounds who might be a little more computational, or like me personally, I'm from more of a like cellular molecular background. And the projects in the lab kind of span computational neuroscience to systems neuroscience to more cellular and molecular type things. So it's kind of a broad use of techniques and things like that. But just kind of like a basic idea of what we study in the lab. So we have a few different projects that focus on different areas of the brain that, such as like the cortex, and also some more subcortical regions like the basal ganglia and the superior colliculus that send these movement commands to premotor regions in the brainstem in order to initiate movements. And particularly in our lab, we use mice as a model animal, and we look at the control of directional tongue licking. So we use some tasks and some behaviors that I can show you guys in a little bit. And so we study mainly how the brain controls directional tongue licking. Let's see. So I guess I can start walking around the lab and show you guys some of the techniques that we use. So let's see. Can't flip the camera, but we're just gonna take a little walk. So here is the hood, pretty standard lab equipment. We use this for things like perfusions and dissecting brains out of the mice that we use, so we can slice them for histology things and check the expression of different viruses that we inject, or things like that. Here is a vibratome that we use to do said slicing. Now we'll walk over to one of our behavior and ephys rooms. So we have this little rig. Let's see if you guys can see it, but basically we have this task with mice that they're head fixed, and they have two spouts that will go in front of their mouth, and they are water deprived, so they're very thirsty, and you can easily train them to learn tasks with water being the reward, because they'll learn they want the water because they're thirsty. And so they're head fixed, and in the particular task that we study, there are two sounds that we can present, and one of the sounds will indicate that the mouse has to lick to the left, and the other sound will indicate that the mouse has to lick to the right. And after those sounds are played, there's a brief delay period in which the mouse cannot lick. And then there's a third go cue that tells them, okay, lick in the direction that was indicated to you by the tone. So that's kind of the basic task that we use. And so you can study, neural activity during the delay period, so motor planning and things like that, and kind of the decision making to go left or right. And then also more like motor commands for the actual initiation of the movement. So that's kind of the basic thing. And so the mice will go in here and get head fixed, And then we also, for this rig, use this for ephys, so we'll use these silicon probes or neuropixels probes that we can insert into the brain and do like large-scale extracellular recordings, and then look at the neural activity in motor-related brain areas and see kind of how they're controlling the licking movement and things like that. Let's see. When a mouse is in there, you can see them at this location, and you can see them doing their, directional tongue licking thing. It's pretty cool. Let's see, now we can go over to our surgery room where we have, now we have Erin, who is a grad student in the BME department. So that's the interdepartmental kind of aspect to GPN, which is really cool. So she is doing, she's doing a surgery right now for probably doing some virus injections and maybe putting an optical fiber over probably one of the brainstem regions that control the directional tongue licking, so that we can do some optogenetics. So that's another thing that we do a decent amount of in the lab. So optogenetics, a lot of virus injections to express different excitatory or inhibitory opsins, so that we can manipulate brain regions that are involved in movement. Let's see what else. So this is another room that we have for behavior. So you can kind of see we have these 3 different rigs set up, and let's see. So you can kind of see maybe in here, I'm not sure where you can entirely see, but basically it's the same thing as in the other room where you have the mouse will be head fixed, and here's where we kind of train the mice to do different tasks. And then you can kind of just sit in this room for a few hours and watch 3 mice at once on TV. It's great, very entertaining. Let's see what else we have. We have another project in the lab that's being spearheaded by a really talented postdoc, and she has to use live slices. So she'll take the brain out of the animal, and she won't fix the brain, she won't perfuse it. And so she has to kind of keep these slices alive so that she can do patch clamp recordings and also some voltage imaging, which is really cool and kind of a newer technique. And so this is the setup that she uses for that, which is quite impressive and tricky, as I hear. Let's see. Here we have just some classic, lab benches for things such as mounting brain slices, so that you can image them, do some histology, like immunohistochemistry or in situ hybridizations, things like that. Here's the area that no one is supposed to touch, and you have to be very careful because you can't have anything that will degrade RNA in that space. So I try to avoid it because I'm kind of clumsy and can contaminate things. And I guess the last room that we have is, so this room has this really fancy, extremely fast confocal microscope that we use for voltage imaging in those live slices I was talking about. So that's really cool. There's a really interesting project that is going on in the lab studying the in vivo connection between different cell types in the motor cortex. So so we do some more systems level extracellular recordings and spike sorting and looking at things like that. And then this other project that we have going on is looking more at the molecular cell types that are in the motor cortex and how those basically talk to each other. So kind of a wide spread of things going on in the lab. And then I personally am not computational, so I don't know a ton about the computational stuff going on in the lab, but there definitely is some. So once again, I'm Jackie in the Economo Lab. It's a pretty good place to be. And if you guys have any questions, feel free to contact me. I'm sure you guys will be given emails or something of current students. And enjoy the interview process, and good luck.